Andrew Fratoni. I'm an Associate Director of Clinical and Laboratory Studies at the Center for Anti-Infective Research and Development at Hartford Hospital in the United States. And this is my poster today. So the genesis of this work stems from the fact that there really is no standardization in how preclinical murine models are run between laboratories across the world. And so Carbex has supported our lab in working with an acronym called COMBINE, which is a group from Europe that has really looked to harmonize and standardize many of the major components of the murine pneumonia model. And so we've been tasked with looking at a collection of Klebsiella and Pseudomonas isolates and establishing a benchmark in terms of quantifying expected CFU per lung for these isolates against humanized exposures of 5 different antibiotics, one of which is tobramycin, which is what this poster is about. So as we know that there's interspecies differences between the lung penetration in mouse and man, so even if you equally match the plasma exposures in mouse to those that you see in man, it does not necessarily mean that at the target site of action in the lung, the epithelial lining fluid, or ELF, that you're also going to get a matched exposure, and you may in fact be either under or overexposing at the target site of infection when plasma profiles are matched. And so when we look to humanize exposures in the lung in mouse, we have to look first at the human data. And unfortunately, the ELF profiles in humans are often poorly characterized by inadequate sampling schemes and extensive variability. And one thing that needs to be taken into account when you're looking at target site exposure is a term called system hysteresis. And system hysteresis describes both the rate and the magnitude at which drug moves from the blood into different body sites, such as the lung. And so if you just look at one point in time, what we call point-to-point penetration, if you look here at figure 1, you can see that over time, for tobramycin in humans, the ELF penetration ratio actually increases over time. So if you just look at one point in time, what we call point-to-point penetration, right, you could misrepresent the amount of drug that's actually getting into the lungs in humans. And so a better way to characterize ELF penetration is the overall exposure, or what we call AUC, of the ELF relative to plasma. And so we looked at the human literature and we found across one study 4 different point-to-point penetration ratios. But ultimately we need to come up with a total exposure. So we were able to create this regression curve as a correction factor for the plasma exposure, which is seen here by the red line in humans. And in doing that, by fitting a linear regression, we were able to come up with a physiologically plausible human exposure of tobramycin in the lungs, which is shown in figure 3 here by this red line. And so we first characterized the plasma exposure in the mice for tobramycin with a 4-dose regimen, as you can see here, which nicely recapitulates both the free Cmax and the free AUC for tobramycin. However, in doing that, so the ELF profile of the plasma HSR was not representative of this red line here, and that's what we don't show you. So what we had to do was make multiple adjustments to the plasma HSR, or human simulated regimen, so that we could approximate the ELF regimen in mice. And so you can see here that the profile of the human ELF is very blunted in the ELF relative to plasma based on the red line. And so it actually took us more doses, but smaller doses in the murine model to approximate the epithelial lining fluid in humans. And so in conclusion, we used a linear regression of point-to-point plasma to ELF, penetration across the dosing interval to determine ELF concentration time profile of a human dose of tobramycin, to come up with an AUC to ELF over AUC to plasma of approximately 0.7. And going forward, it's very imperative for novel compounds that are looking to have an indication for pneumonia that they really highly characterize the ELF concentrations in humans so that we can have an AUC to AUC penetration ratio, which can assist with the preclinical in vivo pneumonia models. So I'd like to thank my mentor and the principal investigator, Dr. David Nicolau, for his support on this project, as well as Carbex for supporting this work. Thank you.